Welcome. I'm delighted you came to join us. I have the pleasure of having Professor Roger Barker here, my hero and mentor from my times in Cambridge, and really I think a visionary person in the field of Parkinson's who's doing a lot of work really in terms of modifying the disease. We try and avoid using the word cure because it's a four-letter word, but we try and talk in those terms. So Roger, we've got the pleasure of having you here in Australia to ask you, pick your brains about the field, where we think the field is going in terms of treating Parkinson's in a disease-modifying way. First, tell us what it means to sort of modify the disease, because people know what the disease is like. What are we aiming for?
Yeah,
cure would be great, but
no, I mean it's a good question, because of course everything modifies the disease at some level. But what people mean by disease modification is a drug which actually gets into the very core of what's going wrong in Parkinson's disease. So in Parkinson's, as you know, these proteins aggregate in the cells become unwell, and then the cell dies. So disease modifying would be something that stops the cell becoming unwell and dying, and a cure, as you say, would be something we gave someone and nothing happened.
Right.
Or it all just went entirely back to normal. So, you know, when you get a chest infection, you're given antibiotics. When you finish the antibiotics, the chest infection is gone, you're cured of it. There is no evidence that that chest infection is there and it won't come back. So cure, I think, is a great thing to aim for, but the chances of getting it, I think, are relatively small in the near future. But if you consider disease modification as a way of slowing down the disease, say, by 50%, you know, so it took twice as long to get to where it was going to get to as it does now, then for most people that would be a cure, because the average age to get Parkinson's is, you know, late 60s, early 70s.
Yeah, and I suppose when people are probably familiar with when they present, they've already lost you know, a large proportion of cells. Most of the treatments we're going to talk about, do you think that they're going to bring those dead cells back to life, or is it gone is gone?
Well, so I think by the time people present, you're absolutely right. You've already lost, say, within one particular area, the dopamine cells, you'd have lost about half of those. And within the half that are remaining, there will be a large proportion that are very healthy, and there will be a proportion that are a bit sick.
Yeah.
So I think if you target treatment for disease modification or rescue at the time when people present, you will rescue a population of cells that are unhappy. You will not, the cells that have gone, and you won't be able to bring them back to life. So obviously one of the big quests, as you know, in Parkinson's is, can we pick things up a bit early? Can we pick it up before people present? And that's of course quite difficult, because if you want to pick up things before they present, then how do I find that? Because do I go out to the population and ask everybody?
Screen everybody.
Yeah. And of course, you know, there are lots of screening programs we have, you know, for different types of cancers, but of course there's not much point screening for Parkinson's disease in the population, because it's a little bit rarer than a lot of the common cancers, but also what can we do about it at the moment?
So right now we don't have anything to offer.
No.
Okay, well, in terms of the disease modifying approaches that we're aware of so far, you mentioned about this abnormal protein which may or may not be killing cells. It seems to be associated with cell death. What are the sort of strategies around that protein, and what are the thoughts about how you deal with it?
So you're absolutely, so there's this protein which is called alpha-synuclein. So that protein, for some reason which we don't understand, people with Parkinson's tends to go into a toxic form, and then it aggregates and it kills the cells. And it's probably not the aggregates that kill the cells, it's probably the toxic form before it forms that. Now that protein exists in everybody's brain. So whether you've got Parkinson's or not, it makes up quite a substantial part of the brain. So if you simplistically say, well, let's just get rid of that protein, let's get rid of it altogether, then there's a good chance if you got rid of it altogether, your brain wouldn't function at all.
Right.
So you can't take that sort of simple approach. So what people are thinking is, can we in some way cleverly identify what's wrong with the alpha-synuclein in Parkinson's disease, and can we target it? And so the simplistic way to think, how could I stop the disease knowing that, is one is you could reduce or stop the production of that abnormal protein. So can I switch it off? And the two ways that people are doing that, one is you can actually do some sort of genetic manipulation with a various agent you can put in to try and switch it off. The other is a very recent discovery that there are some drugs common drugs, so drugs which are used for asthma and other treatments like that, which have the capacity to regulate how much of this protein you actually produce. So one way would be to switch off the production or reduce the production of the protein. The other would be to say, well, I've got the abnormal protein, can I get at that and kill it off? And that's what people are now thinking about, and they're in clinical trials even, with these vaccines or immune therapies. So the idea is you coax your body, or persuade your body by giving you an injection to produce an antibody, which you normally do to get rid of bugs. And here, you're going to produce an antibody that's going to target and specifically kill the abnormal protein that seems to underlie Parkinson's disease.
And those trials are going on now?
So there are at least 3 or 4 companies that are working in this space.
Yep.
And I know of at least 2 that have published trials.
Yep.
Most of the trials, as you can imagine at this stage, are in very early stage, and really what they're testing is whether it's safe. So
right.
if I give people a vaccine,
yep.
are they actually all right? Because in the world of Alzheimer's disease, when they did this, they did run into some major problems with vaccines and inflammation in the brain. So people are wary of rushing ahead because they want to make sure it's safe. And what we can say is in the trials that have been published to date, the 2 trials, it does seem very well tolerated. So patients don't seem to run into any major problems. Whether it makes a big impact, that's to be found out in the next few years.
And we have seen that sort of approach using Alzheimer's disease, and it hasn't been very successful, I think.
Well, it hasn't been successful in Alzheimer's, and I think there are 2 reasons for that, really. Well, 3 reasons, I would say, actually. One is, coming back to a point you made earlier, it was probably given too late.
Right.
So by the time they were using it, people had advanced to quite, well, in the later stages of Alzheimer's disease. So getting rid of the protein would be fine, but all the cells have disappeared. So
you're taking the gravestones out of the graveyard, and yet the bodies are in the ground.
Yeah. So I think that was one of the problems, and there's been a recent trial where they've gone for an earlier stage, and it seems to be more encouraging. So that's the first thing in Alzheimer's disease. The second problem they found in Alzheimer's disease comes back to something we just mentioned as well, which was that when they gave some of these treatments, obviously if you're getting rid of things by producing an antibody, then your body normally, when it gets rid of something, you get a bit of inflammation. So in some people, the antibody was very effective at getting rid of the protein, but it produced a massive inflammation in the brain, and that caused problems. And the third thing in Alzheimer's disease is there are 2 proteins that cause the disease. So if you target one, you have to be absolutely certain that's the only one causing the problem, and it isn't.
Right.
So that, I think, has been why in Alzheimer's disease the results have been disappointing. But the important thing is that people have learned. So these points, it's a bit unsafe, so we've modified how we give it. Going late in the disease is probably not sensible, so they're going early. And thirdly, there's another protein, so now they've developed an antibody against that. So I think, one of the things people, I think, sort of sometimes don't quite understand is that medicine is all about learning as you go.
Yep.
Very unlikely that first trials will ever get it absolutely right and solve the problems. But don't give up, because you can learn and move forward.
And you mentioned the recent paper looking at how you might look at the adrenergic system, the beta system, where you mentioned that effectively drugs that might block that system, like drugs like Ventolin or the beta blockers, the beta agonists, as opposed to the beta blockers that people take for blood pressure and pulse, they would have to be used presumably before someone actually got the disease, so it's going to be a bit harder. So if people have actually got the disease and they're happy to be on those drugs at the moment, is there anything at the moment they should be thinking they should do, or?
Well, I don't think so, because I think this is so new, this discovery. We have to wait to see how it's going to pan out in terms of replication and other things. I think the exciting thing about this paper, which appeared right at the end of August of this year, was that the beta. What they showed in this particular study was that if they looked at a large population of patients in Europe, then those patients who were taking drugs for asthma, which are, as you say, these beta agonists, if you looked at that population as a whole, they had a reduced risk of Parkinson's.
Of getting Parkinson's.
Yeah, so there was less Parkinson's in that population, as though taking these beta agonists somehow reduced your chances of getting it. Yeah. And conversely, if you were blocking that receptor, which is what they call a beta antagonist, it increased your risk. Now, there may be lots of explanations for that, but then they did various studies in this paper to show that beta receptor, so where these drugs were working, also drives how the protein is produced, this alpha-synuclein protein. Now, if you take that forward, what one can imagine is, well, it can obviously reduce people's risk, and as you say, should you take it before you get the disease? If it's a very safe drug, like a beta agonist, like an inhaler, you might say, well, perhaps everyone should take it, but is that going to be a useful way forward? If it also works by modifying the production of this protein, it probably would also help in people who have established disease. But I wouldn't encourage people to rush off to their doctors and say, I want these medications, because it's got to be establish that this is actually true with other people verifying what's happened.
I mean, the other thing that's amusing about this is I'm reminded of the statistic out there that people who smoke cigarettes are at a lower risk of getting Parkinson's disease. And you think, well, are they being given a puffer? Because their lungs are terrible. And actually, we've now found out why smoking might be the issue. I'm not sure what the answer is.
No, but it's a good point, because you wouldn't go out and
start smoking.
You wouldn't encourage the population to go smoking to reduce their risk of Parkinson's disease because of the side effects related to smoking. And similarly, whilst beta agonist inhalers don't really have much in the way of side effects, you're absolutely right. People who get asthma or have chest diseases, for some reason, maybe the very people who don't get Parkinson's, for some genetic reason, they have a genetic background that makes them more likely to get chest disease and not Parkinson's, and it's got nothing to do with the drugs at all.
So one of the other things that's really hot, I suppose, in the field of what causes Parkinson's is the idea of autoimmunity and inflammation, which are kind of related terms. So are you able to take us through what we mean by an autoimmune process driving Parkinson's, and then how that might link in with inflammatory processes?
So autoimmune is a description of a condition where your own immune system or your immune system reacts to something in your own body. So an example of this would be rheumatoid arthritis, inflammation in the joint. So in rheumatoid arthritis, for some reason your body gets confused between producing an immune response to something that's coming into the body and something that's already there. So in rheumatoid arthritis, you'll produce an antibody and immune response that recognises your joints as being foreign, attacks your joints, gets swollen joints, damage to the joints and such like. So that's autoimmune. Your own immune system is attacking your own body. Now in Parkinson's disease, this has always been a suggestion that perhaps what happens in Parkinson's disease is your body gets confused and produces an immune response that kills off the cells which then die in Parkinson's, like the dopamine cells. Now the evidence that it's autoimmune, so if I gave people drugs, which you would for rheumatoid arthritis, you'd stop Parkinson's, that's very unproven. And the idea that the whole disease is triggered by your immune system getting confused between normal brain cells and abnormal brain cells and driving disease, I think there isn't a lot of evidence for that.
No.
But what there is which has been known for many years, is that when cells die off, for any reason, whether it be Parkinson's, Alzheimer's, whatever, there is inflammation associated with cells that die. So if you cut yourself, for example, you will be damaging cells, you get inflammation, we're all very familiar with that. Now, for many years, people said, well, that's just the consequence of losing cells. So when cells die, you get inflammation. What would you expect? I think what's emerging now, coming back to this idea of autoimmunity, is that in some people, this is not actually the simple story, that inflammation is contributing in some way to the disease. So the way one can think about this is that we all respond to things dying, cells dying, but some people react much more vigorously to it. So if you're one of those people, you can start to imagine you get a bit of Parkinson's disease, cells start dying off, your immune system says, oh, the cells are dying, I've got to produce some inflammation to mop up those cells. That becomes very florid, so you get a lot of it. That then damages cells in the area, more cells die, that causes more inflammation, and so it feeds off each other and the disease gets much more aggressive in its behaviour because it's the inflammation that's driving it. It didn't trigger the disease, but once the disease has been triggered, inflammation is important in making it go much quicker. And that, apart from conceptually being very interesting, obviously opens up real therapeutic potential because, as everyone will know, we have lots and lots of drugs to treat inflammation. People take lots of drugs for joint pains, back pains, all sorts of conditions. And so if this is true, then the capacity to take a drug which we already have in the clinic and use it in people with Parkinson's becomes very easy. But of course that's assuming we can show that inflammation really is in some way driving the disease itself.
So again, there would be no advice at this point for patients who've got Parkinson's to go out and start taking these drugs. Presumably they have side effects, I guess.
Yeah, so I mean, it's a very good question and it's a very good thought, because a lot of people ask me this, should I go out and take some Brufen or ibuprofen or whatever, some of these non-steroidal indomethacin. You're absolutely right, these drugs are not innocent drugs, and the major side effects, they can cause major problems in the stomach and cause people to develop ulcers and bleeding and such like. So until such time as we have more convincing evidence, I would never encourage anyone to go and just take these agents on the grounds that they may be helping their Parkinson's.
Okay. An alternate theory that people have, and I suspect that probably the truth behind Parkinson's is a little bit of everything, but a lack of energy in cells. So people might be aware that within the cell you've got the nucleus, you've got other things, and these little power batteries, the mitochondria, which many people have said, well, if we just get those little mitochondria to produce more energy, maybe we'd stop cells dying. Is there anybody looking at that in this space?
I mean, people are very interested in this, and the advantage, why people get very excited about this, especially in the world of therapeutics, is that mitochondria, these little powerhouses within the cell, as you say, if they go wrong in Parkinson's, they probably go wrong in every disease. So a therapy that I could develop to target mitochondria that might help Parkinson's would probably help Alzheimer's disease. It would help any number of diseases. So the therapeutic potential is huge. Now, people have been very interested in this in Parkinson's because for some reason the mitochondria do seem to be selectively involved in the disease process. The critical question is, how much is it involved in the initiation of the event? So is it something that if I target will stop the disease, because that's where it begins, or is it that there's lots of problems with inside the cell with the protein causes other problems, those problems then feed into the mitochondria, and then we get problems with the cell becoming unwell and dying. And if that's the case, then targeting the mitochondria is fine, but all the problems are upstream of it. So all you're ever doing is just holding things for a little while before it gets overwhelmed. So people are very interested in this. Lots of people have thought about it over the years. At the moment, the problem is trying to target a therapy that specifically gets to mitochondria, gets to all the mitochondria, and has the effect that you want. Because obviously these little powerhouses in the cell are pretty vital for how the cell works. So you've got to make sure you get it right. Because if you got it wrong, you could not only not make Parkinson's better, you could make everything worse.
And people have reported on the brains of people with post-mortem samples of Parkinson's and reported things like increased iron levels or changes in copper levels. They're acting not because of the toxicity, or they're acting because of how the cells work. Have people looked at that as a cause of Parkinson's or a way of treating it?
So this is it's a complicated area. It's a bit like the mitochondria, because definitely when you look in the brains, as you say, you have these abnormalities. The question then is like inflammation. The question is, are they in some way responsible for the disease, or are they simply a consequence of having it? Because if they're a consequence of having it, then treating it isn't going to make any difference, because it's just the bystander, it's something that's happened as a result of disease. If in some way it's responsible for initiating and driving disease, then targeting it would be a very good thing. Now, there are drugs which are available for getting rid of things like excess iron, because there are some rare conditions where people produce too much iron, because obviously iron is very heavily involved with the blood, and so there are certain conditions where people produce too much red blood cells, or the red blood cells are abnormal, or you have problems transporting iron around the blood. And so there are agents out there, and they are now going to trials to see if they help in Parkinson's. Now, one of the advantages of targeting that particular area of Parkinson's is not only is that, this is an important question to answer, but you can actually look at iron levels in the brain in people when they're alive, because we can do the magnetic scanning, the MRI scanning, using special sequences on the scanner. You can pick up iron within the brain, and therefore you can know if you give a drug that's supposed to mop up iron, if the iron levels in the brain actually go down on the scanner. So you know whether your drug has worked, and then you can ask the question, well, does it make any difference to Parkinson's disease?
And you introduced a topic there of, if you like, repurposing existing drugs. So we have a whole bunch of drugs that are in humans, in clinical practice. Where are we with those drugs and the idea that they might be able to help with Parkinson's? Are there any?
Well, there's a huge move towards this. It's not just restricted to Parkinson's, I should say, but this idea of drug repurposing. So as you say, you take a drug which is well established for one indication, say statin, simvastatin. Well, many people in the world take it for cholesterol, for heart disease, strokes and such like. So this is a drug where we have a huge amount of information on how safe and well tolerated it is. And then the idea is, of course, you take a drug like that and say, would it be useful in Parkinson's? Because in our laboratory-based studies, we've shown that simvastatin, for example, does reduce inflammation in animal models of Parkinson's disease. So if we reduce these inflammation in animal models of Parkinson's, would it reduce inflammation in people with Parkinson's? Would it help them with their Parkinson's? Hundreds of millions of patients have had these drugs, so why don't we just try it in Parkinson's? So there's a lot of interest in this. And I suppose the area where this has proven to be the most exciting at the moment is in a drug which has been used for diabetes. So there's a drug called exenatide, which is one of these drugs called a GLP-1 agonist. And so there's some evidence from 2 trials now that may have some effect in Parkinson's disease. There are a number of other agents which people are trying, and this area has become very active through really the initiative of the Cure Parkinson's Trust in the UK. They've set up a thing called Linked Clinical Trials, where they're trying to really facilitate and allow this to happen. Now, there are several assumptions in all of this. So the first thing I would say is I think the chances that one of these drugs is going to have a major effect is not great, because I think someone would have spotted it by now. So, for example, if simvastatin really helped in Parkinson's, some astute clinician would have said, it's funny how all my patients on simvastatin do so much better. And that's not my experience, and that's most other clinicians' experience. Secondly, there's an assumption that the dose I use of a drug for treating cholesterol, so simvastatin, is the dose I need to reduce inflammation in the brain in Parkinson's, and we don't know that. No. So dosing may be an issue. But the great thing about this and the excitement of it is that it's really caught fire in the last few years. We can do them now. So all we need to do is get the money and the ethics. We don't need to do lots of extensive testing of safety and tolerability. So we can quickly move to trials. Yep. And by coordinating this, so I think this is the other important thing, this isn't the province of Sydney, it's not the province of Cambridge, this is something we can do globally in a much more efficient and better way.
And I think you've been very instrumental in that with your association with Cure Parkinson's Trust, and worth saying that there are efforts in Europe already, and I think Australia is now starting to get these trials running. I think we've not seen much of this, but I think it's great credit to yourself.
Well, I think the other it's very kind of you to say that, but I think Australia's been very receptive to this, and I think it's people like yourself who've come here and prepared to engage with it. So I think one of the difficulties in trying to do things internationally is that people can somewhat retreat into their own country's culture rather than looking out to say, can we do this in a more global way? And ultimately, there's no reason that Parkinson's in Australia should be any different to Parkinson's in the UK. So if it works one, it should work somewhere else. And I think trying to embrace that and make sure that the world community world Parkinson's community is involved with it is very important.
I think it's interesting because Australia has seen itself as being a long way away, and industry has seen it as being not a very accessible place to go and do trials are expensive. But here what we're talking about doesn't really rely on the industry, it relies much more on people making donations in philanthropy and saying, well, okay, we've got willing cohorts of patients here in Australia who are dying to do this, and all around the world who are dying to do this. We just really now need to be able to coordinate ourselves and test the science out in a good way. So I think there's a real spirit of that.
It is, and also I think the other thing is that whilst people say, well, Australia's quite small and it's a long way away. Coming back to what I said earlier, if it works in Parkinson's in Australia, it's going to work in Parkinson's anywhere in the world. So, and I think companies are now recognising that there's no point worrying about one country. I mean, they do worry about America because of the size of the market. Yeah. But fundamentally, if these treatments work, they can be rolled out across the globe.
Well, look, I think it's been an absolute delight having this time to talk to you and hearing all about these exciting prospects. We're grateful for your time, and I know that all of our patients and people tuning into this will be blown away by your knowledge on this, and very grateful for the insights you're able to convey in very simple language. Thank you so much for your time.
Well, thank you for having me.
Pleasure. Beautiful. We love that.